Computational Neuroscience. Definition is the study of brain function in terms of the information processing properties of the structures that make up the nervous system.
Hello everyone, welcome to Computational Neuroscience. I'm Rajesh Rao, a professor of computer science and engineering at the University of Washington.
And I'm Adrienne Fairhall, a professor of physiology and biophysics at the University of Washington.
In this course, we'll embark on an exciting journey into the computational function of one of the most complex and amazing machines in the universe, our brains.
We'll learn how brain cells, also known as neurons, process and encode information, and how we can use neural recordings to decode the meaning of the brain's activity. We'll also show you how to use our understanding of the physics of neurons to simulate them on computers.
We have about 100 billion neurons in our brains. These neurons are connected to each other in intricate networks. What types of computations in these networks allow us to perceive and act on the world? How do our brains learn by adapting the connections between neurons in these networks? These are some of the questions that we'll tackle in this course.
Each week, we'll give you about an hour's worth of recorded video, and you'll also have the opportunity to try out some of the ideas for yourself in a weekly problem set.
We invite you to join us on this 8-week-long journey into the fascinating world of computational neuroscience.
The human brain is the most complex structure that we know of. It processes the input from our senses, coordinates our movements, and is responsible for the multitude of our thought processes. At the basis of all our brain's abilities lies the activity of single nerve cells. Finding out the rules of their interplay is the goal of computational neuroscience. Nerve cells communicate in the form of electrical pulses that are passed along from one cell to the next. These pulses actually encode information, which is then processed within a nerve cell. The result? The cell either reacts by emitting a pulse of its own or remains silent. However, the human brain does not consist solely of only a handful of nerve cells. Rather, an estimated 10 billion of them exist in the cortex alone, with each of these single cells communicating with up to 10,000 of its neighbors. The question is, therefore, how can we investigate and describe these processes in a scientific way? And how does the activity at the cell level relate to what we eventually perceive as the brain's abilities or dysfunctions? In order to understand these processes, it is important to realize one central fact. Nerve cells form a network. Whether we are talking about a network of social interactions, the World Wide Web, or the brain, networks adhere in their functions to certain rules. If we want to understand the network that is our brain, we have to take a look at single nerve cells. Helpfully, the rules that lie at the basis of their behavior can be described in the language of mathematics. By taking this approach, mathematical equations help to explain why a network develops in a certain way, or reacts in a specific way to outside influences. Furthermore, an additional advantage is that once these processes are described as an equation, they are then able to be dealt with by computers. This way, artificial networks can be created that resemble the structure of a nerve cell population within the brain. Today, computers have become powerful enough to provide realistic simulations of neuronal networks and help to understand phenomena that was observed in experiments. Yet still, recreating a complete human brain is a distant prospect. The results of these computer simulations can be compared to insights that neuroscientists have found in their investigation of real brains. The ongoing exchange of theoretical and experimental data is of great importance. It ensures that the abstract models are consistent with reality. Describing the brain's processes by using mathematics and running simulations of networks and computers aids our understanding of how billions of nerve cells cooperate, and how this results in the processing of input from our senses, the coordination of body movements, and the ability to think. This increased understanding will also help us to find causes for certain dysfunctions of the brain. These are the principal goals of computational neuroscience.
Computational neuroscience is distinct from psychological connectionism and from learning theories of disciplines such as machine learning, neural networks, and computational learning theory, in that it emphasizes descriptions of functional and biologically realistic neurons and their physiology and dynamics.
I am John Milton. I'm a professor of biology and the William R. Keenan Jr. Chair in Computational Neuroscience at the Keck Science Center. When I came from University of Chicago to the Claremont Colleges, I was interested in developing a program specific for students at a liberal arts college. And the first thing I noticed is that there was so much movement going on campus. Students are interested in sports, dance, things like that. And so the idea was, well, why not build a research program for undergraduates based on movement? And that's exactly what we've done. The kinds of experiments that we decided to focus on were the aspect of movement that relates to balance control. And so the colleges here have actually become very famous for their work on stick balancing in humans, and have generated 37 publications in the last 8 years with something like 25 student co-authors. Here, every professor knows all their students. All the students get to run all of the equipment, and they also get one, maybe two independent research experiences, so a completely different thing. The second thing I emphasize is that the students get to do things to make their resume look really outstanding, which is important when it comes to the time of medical school or graduate school application. I had one student who, in their study abroad, had done human physiology, exercise physiology at the University of Queensland in Australia, which is the #1 school in the English-speaking world for exercise physiology, and then came back through our REBME program, that's Research Experiences the Biology Mathematics Interface, worked at USC, which is the top kinesiology program in the United States. And when she came time to apply for graduate school, she was fully funded to go to USC to do a PhD in kinesiology and concurrently with a doctor of physiotherapy. That's the kind of student stories that we have quite a bit here.
Hello everyone, my name is Eugenio Colorcello. Today we're going to talk about some of computational neuroscience and learning basic information that one might want to use when studying artificial and robotic vision systems. We will start first trying to figure out why do we have a visual system. You know, our eyes, the eyes of humans have evolved for different reasons, and not necessarily to just look at a nice multi-megapixel picture from your camera phone. Eyes has evolved to study the environment and to figure out important features of the environment. There are sensory structure capable of spatial vision, imaging the environment no matter how crude the image is. As my thesis advisor Andreas Andreas used to say, the most important things about a visual system is just to extract a few bits, a few amount of information in the right place at the right time. Is there something interesting in the environment? In a specific class of object, where it is and what it is? Those are really the question that the human visual system or any other animal visual system that have evolved to really answer. In this class we're going to talk about the principle of computational neuroscience about mainly visual system in humans, but this applies really to any visual system, no matter how crude it might be. So when we talk about the the human visual system, at least for the purpose of this course and class, and for the purpose of artificial vision system, I want to focus on a few key elements. We're gonna take a look at eyes that have some specific property to report the visual information to the brain. Usually, the brain can be divided into at least the visual cortex, which corresponds, is thought to be about almost a third of the entire human brain. Has many different areas. We're gonna focus on the one that are in blue here on these slides. But as you can see from diagram from Simon Thorpe, retina takes input. After 20-40 milliseconds, it passes them to another nucleus in the brain, the lateral geniculate nucleus, which takes another 30 to 50 milliseconds later, and passes the information to the primary visual cortex, which is called V1. Then this information is passed to visual cortex area 2. And then, usually the pathway to identify object is flows from V1, visual area 2, visual area 4, which reports intermediate visual features, and we'll see in a few slides. And then it will go into the inferotemporal cortex, IT, where objects in the scene is deconstructed into elements. Only then the brain is going to notify the prefrontal cortex, so the aware part of your brain, about what is being seen. And based on that, you might decide to do some motion command that control some muscles in your body. But it could stop at the prefrontal cortex just to be aware of the environment, for example. So in this course, we're going to look mainly at eyes, and then this pathway, which is the ventral pathway of the visual system that sort of recognize objects, which is the most important thing that we're going to study in this course, really. So I wanted to give you some information and some ideas about how our brain and nervous system can basically compute features that allow us to understand what the world is about. Before we delve into that, I want to give you a tool that is some kind of words that are used very often in neuroscience, and also now in artificial vision and robotic vision, because many of these algorithms are inspired by neuroscience. This tool is the receptive fields. So what is a receptive field? I'll tell you in a very the simplest way I can say. So receptive field basically are an aggregate of information. If you think about a bunch of cells, that could be any kind of cells. These cells are going to get some specific input. For example, this could be neurons in your eyes, or detectors in your eyes, and each one of them is going to see something different, but they're going to pass the information to one or multiple neurons, let's say one. So the way they pass this information is aggregated together so that it gives an input to the next neuron, the next layer. It's called a receptive field. So what really this reminds is that each one of these neurons is connected to another neuron in the next layer in a certain way. So these lines will have a certain strength. There's going to be an operation here that is performed because none of these things might be equal. If the input were all equal, this would correspond to some kind of averaging, but it doesn't have to be, as we'll see. So what this operation does, what we refer with receptive field, is basically this neighborhood operation where you take a bunch of inputs, a lot of inputs, and this is really the receptive field of this neuron, is whatever neurons here can change the input to this neuron. This is the receptive field. You can think of it as a local vote or a sampling where this neuron samples from all of these other neurons and receive an answer, or this neuron, all these neurons here in the neighborhood can vote to basically propagate some information to the next layer. And receptive fields are everywhere in sensory processing areas of the brain and the nervous system. There are receptive fields in your eyes. So for example, if you're seeing a line is composed by a bunch of dots, for example. These dots could, there are neurons, for example, that are just able to see dots, but combined together, if we combine together all of these elements, then the next neuron is going to be able to see a line, because it sees whether there's dots in all of this, in this area here. In the retina, could be that many photoreceptors are combined together to give rise to one response from one of the ganglional cells, the output of the retina. In other sensory touch, for example, some neurons in the cortex could be sensitive to a vast area of your finger, for example, and that's what is displayed here is the receptive field in different kind of sensory input, like pain, touch, vibration, or heat detectors. When we talk about receptive fields, we should also talk about artificial receptive fields, because we're going to try to emulate those receptive fields in computer programs, and this is done usually by employing an ensemble of neurons as an input. So you could have a matrix that represent values of a neuron or of an image, or some kind of a map, touch map or something. There's going to be a voting scheme that represents basically these connections, which we call kernel or convolutional kernel, which is going to take all this neighborhood here to generate only a single output. So each kernel, each sets of connection generates a single output, but there could be multiple sets of connection that generate multiple output maps. So you see that there's a neighborhood and one output, and there's a voting scheme or a connection scheme that decides, which one of these units have more importance to get the information that we want out here. We often call this convolution operation, which you might remember from your mathematical classes, and I would strongly recommend you that if you don't know what we're talking about, that you really go read about the convolutions again. Convolutions are really, a receptive field for us, and in fact we're going to use them throughout the classes. So I wanted to tell you that in the case of an image, for example, when it's represented in a computer, it's just a bunch of numbers, and so the input image here is this set of numbers that you see here. A convolutional kernel will do some operation that will bring up some response. For example, you might want to find an edge, a line, or some features about that part of the image. And more on this, we will see more of that.
Behaviors of networks. Biological neurons are connected to each other in a complex, recurring fashion. These connections are, unlike most artificial neural networks, sparse and usually specific. The interactions of neurons in a small network can be often reduced to simple models, such as the Ising model.
What I'm interested in is how we see, recognize, and act upon the world, and how our brains make sense of all this very complex information that hits our eyes in order for us to then interact appropriately. And one of the difficulties with explaining this is that it all happens so quickly that we don't really think, what is there to explain? But actually, you get great insights into this by working with people who, after a brain injury, start having problems. The brain can no longer make out exactly what they're seeing. They have problems recognizing exactly what it is or who it is, or they have problems controlling their motor system, their arms, their legs, in order to interact appropriately.
Working with Glyn and the team have sharpened up my reactions. The tests have helped me with my concentration and reaction. Sharpen me up, for want of a better phrase.
I come every week, yes. Good thing for me, I think. Things I couldn't do, I can do now. I used to cash, I couldn't do it. Now I can, no problem at all.
Birmingham University has got great facilities for this kind of research. We've got a research-dedicated MR scanner, which gives very high detailed images of people's brains, both the static picture but also the activity going on in people's brains while they carry out various tasks. We have facilities for measuring brain activity on the scalp using EEG, and we have procedures such as transcranial magnetic stimulation, or TMS, which allow us to directly intervene on the brain by stimulating the brain and therefore helping recovery of function after a brain lesion or a stroke. There are quite a number of intended outcomes for the research. For instance, at the moment we're running a very large-scale trial of looking at the cognitive problems that people have after a stroke. And the outcome there is to develop a clinical procedure for measuring cognition, as we call it, that is, memory, language, attention, and so forth, that will then be applied to patients as part of the standard care pathway in stroke. And our aim there is for that actually to become a standard procedure in the NHS, for instance. Linked to that, we're looking at the brains of the people and seeing whether we can develop means of predicting who's going to do well, who's going to have particular kinds of problems with particular tasks and so forth, so that we can target rehabilitation better. And then finally, we're looking at rehabilitation itself, trying to devise new ways of helping people recover after brain damage.
Cognition, discrimination, and learning. The brain seems to be able to discriminate and adapt particularly well in certain contexts. For instance, human beings seem to have an enormous capacity for memorizing and recognizing faces. One of the key goals of computational neuroscience is to dissect how biological systems carry out this complex computation efficiently and potentially replicate these processes in building intelligent machines. Computational clinical neuroscience is a field that brings together experts to train scientists and clinicians that wish to apply these models to diagnosis and treatment. Computational neuroscientists aid our understanding of information processing in the brain and often work in tandem with experimental neuroscientists to continually refine their models.